So, thank you for the introduction, Anders. Like Anders just mentioned, I will talk about the definitions and give you a very brief introduction to the field. I saw that many of you raised your hands before, so I guess it seems like most of you already know pretty much what it is. But I will give you a little bit more details, and also, for those that didn't know, I will tell you. So my name is Erik Gullberg, and I work as a PhD student at the Department of Medical Biochemistry and Microbiology over at BMC at Uppsala University. So we'll start with some definitions. When you want to know a definition about something new, I usually go to Wikipedia. So this is what Wikipedia tells about synthetic biology. That synthetic biology is a new area of biological research that combines science and engineering. If you read further down, it continues with saying, it puts an emphasis on developing foundational technologies that make the engineering of biology easier and more reliable. And I think these are two key words here, easier and more reliable, because working with biology and genetics traditionally has been very far from easy and reliable. So this is a big change. If you go to a more official definition, the European Commission said in 2005 that synthetic biology is the engineering of biology, the synthesis of complex biologically based or inspired systems which display functions that do not exist in nature. And they continue with, synthetic biology will enable the design of biological systems in a rational and systematic way. And here we're back to the more engineering view on it, that it should be rational and systematic. So you could say that synthetic biology is simply an engineering view on biology. And here we look at cells, instead of being something quite mysterious and difficult to explain, we try to see them as very complex biochemical machines, or kind of like computers. And if you use that analogy, their DNA is their programming language. There is, however, big differences between how a computer would work and how a cell works. For example, in a computer you have a very big distinction between hardware, that is the actual physical object in the computer, and the software, which is the program code. While in biology, these two kind of are the same, since the DNA doesn't only control how the cells handle information, it also controls exactly how the hardware will look. It completely defines the entire cell. And you could say that synthetic biology then represents a transition from working with biology as a science, where you just try to find more knowledge about how the living systems work, to be able to go to engineering, to know so much about them that you can actually construct new things based on these systems. There's been a term around for quite a while that is called genetic engineering. And when you first hear this, you would think that they should be kind of the same, right? And the line between genetic engineering and synthetic biology is not perfectly clear where the line goes. But in general, when you talk about genetic engineering, you talk about some more older technologies, like reading DNA. We've been able to do that for quite some time now, to sequence genomes of organisms and see how the code looks. And also cutting and pasting this DNA, to use restriction enzymes and ligases to cut out pieces of DNA from one organism and paste together with DNA from another organism and make it to work somehow. We've been able to do this for quite some time. So what is new with synthetic biology is that it's much more dependent on also writing DNA, not just copy-pasting things around, but actually writing it just from designing a code and then having it synthesized, just the way we want it, without being so dependent on what was previously there. Also, abstraction and design. I will get back to this, what I mean with this later, but if you want to make complex genetic designs, you need to have some kind of level of abstraction. And also standardization. If you want to make this engineering discipline, you need to be able to exchange parts, and you need to have some kind of standards that you follow. Before, biology has been a bit like industry was, or engineering was before they had standards. Every company used their own parts and their own bricks and their own bolts, and you could never exchange parts between them. But once you have standards, everything is much easier. So to get back on writing DNA, I mean, we've been able to write the DNA to some extent for quite some time. We've been able to synthesize oligos, or short stretches of DNA, but we haven't been able to really make large piece of DNA without it getting way too expensive. But the last 10 years, the prices of gene synthesis have dropped a lot. That's what this picture shows, that this is the cost of gene synthesis, this is the cost of oligos, and this is how the productivity of synthesis generally has increased. And this is a log scale, so this is a massive increase in both how much DNA we can synthesize and how cheap it is. This is a bit outdated, this is from 2009, but the general trend is still going. And what this makes possible is that traditionally when you try to make a genetic design, you have to spend lots and lots of time doing the actual construction, cutting and pasting new DNA together to form the kind of system you want. But when you can if you can just design it in a computer and then order it from a gene synthesis company and then get exactly that construct back in the mailbox a few weeks later, it can increase the productivity of science a lot. And this is also something that makes it possible to do true genetic programming when you're just working in the computer first, you're making your design, and then you just order it exactly the way you want it, without being dependent on what is already there, how can I cut and paste this together. So if you want to do genetic programming, you need to work, you need some kind of abstraction to this. You need to be able to use that when you design. And with that I mean that if you continue comparing to a computer, today no one would program a computer just using binary code, just writing ones and zeros. Even if, actually that's the code the computer runs, it's just binary code. But we cannot write complex programs that way. So we need to use some kind of higher level language to program them. And we're trying to do something similar in biology, in the way that instead of designing our programs on sequence level, just writing nucleotides, we're trying to make some kind of abstraction and turning the DNA into parts. And once we know how these parts work individually, we can put these parts together to form a complex machine. And that's what this is symbolized. These are different parts. It could be a promoter that activates, that tells when a gene should be active. This is a switch, call it. This is a reporter. This is a blue fluorescent protein that turns on under certain conditions. So I'm using the word part now a lot. So what exactly is a standard biological part? There was a definition made in 2008 by Drew Endy, one of the big names in synthetic biology, where he said that a standard biological part is a genetically encoded object that performs a biological function and that has been engineered to meet specified design or performance requirements. And so here, this is very much an engineering view on this. And once you have all these parts and you know how they work individually, you can put them together. And to be able to make a really complex system, you need some kind of abstraction hierarchy. And that means that, like I said first, if you want to make a new part, you need to look at the sequence level. Then you might actually need to look at the actual nucleotides that form this part. And then you make a part. So this is the part. This is a coding sequence that forms a gene. In this case, it's a repressor that works like a switch or a dimmer. It can turn a gene on or off. And once you characterize this part and you know how it behaves, you can use it together with other parts and form a device. So a device is made out of several parts together, and now you can make a more complex function. Here we have parts that together form an inverter or a logic NOT gate. So here, if you have a signal that comes in that is high, you get a signal out that is low. So input 1 will give output 0, and input 0 will get output 1. And when you have these kind of parts, you can make more complex systems. So next level of abstraction is system level. So here we can actually make senders, receivers, inverters, output, by just putting these devices together. And these kind of systems would be extremely difficult to construct if you would be designing them on sequence level. So once you have these parts and you have this abstraction, you can work with some kind of more engineering work. traditionally, biology hasn't really worked that way. But if you compare to some engineering project, if you would build a house or a bridge or something, you would first make a specification. What do we want to construct? Based on that, we can make a design. How should we make this? You can do some modeling. Will this bridge hold? How many cars can we run over this bridge before it breaks? Once that is done, you can do construction. You can construct the actual bridge, whatever it is, and then finally you test it and see that it does what it's supposed to do. And this is where we want to get to in biology as well, that we have these different steps. And if you have this in engineering, you can separate these steps. It's not the same people designing a bridge that will actually build the bridge. And we want to make it the same in biology, that you can separate this instead of decoupling. That it could be one team of researchers or engineers that do the specification, others can do the design, another team can do the modeling, and then when that is done and you see it seems good, you do the construction and then you try it out. Because traditionally in biology, this is not how it's been working. It's usually the same team doing all of these things back and forth a little bit until it works. So back to standardization. When I say standardization, what do we mean? Well, there are several aspects of a standard. One is simply the physical. How do we put these parts together? How do we link these pieces of DNA? The other part is how do we define a function? There are not always units that are for these kind of things, like a promoter that regulates how strong a gene is expressed. What is a strong promoter? How do we define strength and what do we compare to? And also it's important to look at the system they will be working in. Are these, for example, what organism are this system made for? Does it work in yeast, in bacteria, in dogs? What system is it made for? And is it dependent on temperature or media and things like that? So I will go back a little bit to these different factors. So if you start with the physical part, the standards for assembly. When we put the genes together in synthetic biology, we usually call that assembly. And these standards are simply how do we put parts together in a standardized way. And we usually use restriction enzymes. And so the standard is simply how do we define the ends, the parts that are surround this part. And so that you can cut them in a standardized way and put pieces together. And once they're put together, they form a new part and cannot be taken apart again.